Welcome back. Today, we're diving into something pretty groundbreaking, a health data project on a scale we've really never seen before.
That's right. We're looking deep into the blood.
Exactly. We're focusing on those chemical markers, the ones floating around in your bloodstream, your metabolome, and asking, how deeply do these tiny molecules connect to practically every aspect of our health? Diseases, traits, risks, everything.
It's like trying to build the ultimate map of human health, really.
Mm.
And this study leverages these incredible long-term resources, specifically the UK Biobank.
Huge resource.
Massive. And they've created this, well, atlas. It gives us this dynamic, almost real-time snapshot of what's happening inside us.
Okay, let's start with the star of the show, metabolites. For anyone listening who isn't, deep in the biochemistry weeds, why are these molecules so important? More significant than, say, just looking at our genes?
Great question. Think of metabolites as the most immediate readout of your health or disease state, for that matter.
Readout?
Yeah, like a printout. Your genes, your genotype, they're like the instruction manual. They tell your body what it could do, the potential.
Right, the blueprint.
Exactly. But metabolites, they're the results. They are the actual end products of all the cellular processes happening right now. They reflect your diet, your environment, your lifestyle, and your genetics all playing together.
So they capture everything happening now.
Instantly. That's why they're so much more closely tied to what we can actually observe your phenotype than other biomarkers. They tell the story of, like, the last meal you ate, but also potentially the next 10 years of your health.
Okay, so our mission today is to navigate this new human metabolome-phenome atlas. This is built on data from, get this, 744,241 people in the UK Biobank.
And followed for a long time.
Yeah, a median of nearly 15 years. We're basically sorting through, over 1.4 million potential links between these metabolites and health outcomes, trying to find the really critical stuff.
The scale is just staggering. They systematically mapped 313 specific metabolites in the blood plasma measured using NMR, that's nuclear magnetic resonance.
Okay.
And they link these to over 1,300 diseases and more than 3,100 different traits. The final tally was over 52,000 significant metabolite disease connections alone.
Wow. Okay, so with almost 15 years of data, how did they even start to organize that, especially with diseases popping up at different times?
Right, that's a key methodological point. They had to distinguish between two types of diseases. First, prevalent diseases.
Meaning
Diseases people were diagnosed with before their blood was drawn for the study. That gives you a cross-sectional view, a snapshot of who was sick then.
got it, like a baseline.
Exactly. But then, crucially, they looked at incident diseases. These are the ones diagnosed after the blood sample was taken.
Ah, the predictive part.
Precisely. They use methods like Cox proportional hazards regressions, which is basically the gold standard for figuring out future risk over time. This incident analysis is what really gives the study its crystal ball power, so to speak.
Okay, let's talk big picture findings then. If you had to pick one metabolite that was like the biggest villain associated with the most different health problems, what was it?
That dubious honor goes to a specific lipid ratio. It's the ratio of cholesterol to total lipids within large, low-density lipoprotein particles, expressed as a percentage. We call it LDLC percent.
LDLC percent. Okay, a specific type of LDL cholesterol measure.
Right, a measure focused on the large LDL particles. And this single measure was associated with an incredible 526 different diseases.
526? Seriously?
Yep. It just hammers home how central this particular kind of lipid problem dysregulation is to, well, system-wide disease.
That's not just heart disease then, is it? 500 diseases. It sounds like it's linked to almost everything going wrong, like a fundamental failure point.
You could definitely see it that way.
Yeah.
Now, where did these associations cluster most strongly? For the prevalent diseases, the ones people already had, the highest proportion of links were found in hematologic and immune diseases, so blood and immune system disorders, about 31.8%, followed pretty closely by endocrine and metabolic diseases like diabetes at 28.6%.
Okay, so existing conditions strongly tied to blood, immune, and metabolic function. Makes sense. But what about the future risk? The incident findings. That's the really fascinating part.
That's where things shift. For predicting future disease onset, endocrine and metabolic diseases jump to the #1 spot accounting for 26% of the incident associations.
So metabolic problems predicting future metabolic problems mostly.
Right. But interestingly, number 2 for future risk was infectious diseases at 24.1%.
Infectious, that's surprising. How would metabolites predict catching something?
It likely reflects underlying immune function or susceptibility, which is itself influenced by metabolic state. It's complex, but suggestive.
Okay, let's pause for a quick recap here. We've got this incredible map linking 313 key blood molecules to thousands of diseases and traits over 15 years. We know specific lipid ratios, like that LDL-C percent, are major players linked to hundreds of conditions.
A central hub of risk.
Right. So, for you listening, what really stands out about where these links are strongest? Why do you think metabolic disorders seem to dominate the predictions for future risk, the incident findings? It really feels like metabolic health is the first domino, doesn't it?
It does. And if metabolic failure is the first domino, perhaps the most profound insight from this whole study is just how early that domino starts to wobble.
Okay, you mentioned the predictive power. How far back are we talking? How early can these metabolite readings act like a time machine for disease?
This finding is honestly pretty stunning. Over half of 57.5% of all the metabolite disease pairs they identified showed statistically significant differences in the blood moving forward, over a decade before the disease was clinically diagnosed.
10 years.
10 years.
That's almost hard to believe. A decade before symptoms even appear, your blood chemistry is already sending out warning signals.
That's what the data shows. It's potentially a massive window for early intervention.
Okay, was there one specific molecule that acted as the most common early warning siren across many diseases?
Yes, the ratio of triglycerides to total lipids, again those large LDL particles, LDL-DTG percent, this measure showed these early pre-diagnostic variations in 246 different diseases.
Another large LDL measure, triglycerides this time.
Right, it's a huge flag for identifying risk long before symptoms. And another really interesting finding related to timing, specifically within the 5 years leading up to diagnosis, changes in the degree of unsaturation in fatty acids were linked to the most diseases showing variation.
Unsaturation, so the type of fats in your system, like related to diet.
Exactly. It suggests the chemical nature of the fat circulating in your body starts to look unhealthy years before the disease really takes hold structurally.
Wow, okay, if these signatures are showing up so early for multiple diseases, it makes you wonder if different diseases share similar metabolic weaknesses, right? Do they kind of cluster together based on these early warning signs?
They absolutely do. The researchers used the temporal patterns, how metabolite levels changed over time before diagnosis, to group diseases into 44 distinct clusters.
Sort of like a metabolic comorbidity map.
That's a great way to put it. It groups diseases that might share underlying metabolic pathways going wrong.
Can you give us an example, one cluster that really highlights this?
Sure. Take cluster 1. On the surface, it includes a mix of conditions. Metabolic disorders, yes, but also cardiovascular problems, and even things like auditory impairment, hearing loss.
Hearing loss, what's the connection?
The shared thread seems to be underlying mechanisms like lipid dysregulation, there are those lipids again, and atherosclerotic processes, the hardening of arteries. These processes affect tiny blood vessels everywhere, including the ear.
Ah, okay. Makes sense.
And a key marker for this cluster was the ApoB-A1 ratio. This ratio showed consistent differences compared to healthy individuals throughout the entire 15-year period before anyone in that cluster was diagnosed with any of those conditions.
So you're charting the shared roots of diseases we often treat as completely separate problems. Fascinating. What about aging? Does our metabolic signature change predictably as we get older?
It does, but interestingly, not always smoothly. They used a specific algorithm called DSWAN to analyze age-related changes. It found 2 distinct crests, or periods, where metabolic changes really accelerated.
When?
Around age 46, and again around age 64. It suggests that, metabolically speaking, aging might happen in these sort of waves, rather than just a steady decline.
Mid-40s and mid-60s. Interesting points of transition.
Yeah, and they grouped metabolites based on how they changed between 40 and 70. Some showed steady increases or decreases, but others had more complex nonlinear patterns, like rising until about age 60 and then leveling off or even declining.
Okay, and finally, in this section, something you hinted at earlier, the crucial need for personalization. This isn't a one-size-fits-all picture, is it?
Not even close. This is a really critical takeaway. Nearly half of all the significant associations they found in the entire atlas were unique to specific sex or age subgroups.
Half. So A finding might apply to older men, but not younger women, or vice versa.
Exactly. It demolishes the idea that we can rely solely on universal risk factors or biomarkers. Biology is highly context-dependent.
Give us the most striking example of this divergence, the one that makes you go, whoa.
Okay, look at liver disease risk and certain components of high-density lipoprotein, HDL, often called the good cholesterol.
Right.
The study found that specific HDL measures, like the size of the HDL particles and the amount of free cholesterol in large HDL particles, LHDL-FC, were strongly protective against liver diseases in females. Higher levels meant significantly lower risk for them.
Okay, protective for women. What about men?
For men, the exact same metabolites were associated with increased risk of liver disease.
Wait, seriously. Protective in one sex, harmful in the other.
Exactly. The biological meaning of that molecule completely flips, depending on whether you're male or female, at least for liver disease risk in this dataset. It's a stark reminder of how different male and female physiology can be at the molecular level.
That's incredible. Okay, another quick summary point. We've learned metabolites can signal trouble potentially a decade early. Diseases cluster based on these shared long-term metabolic vulnerabilities. And crucially, factors like sex and age can totally change whether a metabolite is good or bad for you.
Huge implications.
So thinking about that liver disease example, HDL, being protective for women but risky for men, it really begs the question, how do clinical guidelines need to change? We can't keep using these blanket universal risk factors if they behave so differently in different people, right?
Absolutely. The future has to be more personalized. Which leads us nicely into the next part. Can we actually use this information for prediction?
Right. The study didn't just map associations, they tried to build predictive tools. They used machine learning to create something called a metabolic risk score, or MetRS. Given they started with 313 metabolites, how complex was this score?
Surprisingly simple, actually. That's what's so powerful. They found they could get excellent predictive performance using only the top 30 most important metabolites. That's less than 10% of the initial markers they measured.
Hold on, they basically threw out 90% of the metabolite data and still got good predictions with just 30 markers.
Exactly. This simplified 30-marker MetRS achieved really strong classification performance in area under the curve, or AUC, above 0.8 for 94 prevalent diseases and 81 incident diseases.
And AUC 0.8 is generally considered good to excellent, right?
Very good, yes. For context, predicting prevalent type 2 diabetes using this MetRS hit an AUC of 0.941. That's incredibly accurate at distinguishing who has diabetes from who doesn't, just based on those 30 metabolic markers. For incident T2D, predicting future onset, it was still very strong at 0.863.
Amazing. Were there any specific metabolites among those top 30 that just kept showing up as important across lots of different diseases, like universal power players?
Yes, 4 markers consistently rank near the top for predicting both current disease and future risk: creatinine, glycoprotein acetyls, glycine, albumin, and acetate.
Glycine, I've heard of that one. Isn't it linked to inflammation?
That's right. It's considered a pretty robust marker of systemic inflammation. And in this study, it was a strong predictor for diseases as varied as lung cancer and the autoimmune condition Graves' disease. Creatinine, often associated with kids. Sure. In simple terms, MR uses naturally occurring genetic variants that are known to influence the level of a specific metabolite as a kind of natural experiment. Since your genes are assigned randomly at conception, if people with a gene variant for higher metabolite levels also consistently have higher rates of a disease, it provides much stronger evidence that the metabolite itself is causally involved in the disease, not just correlated.
Got it. So it helps untangle cause and effect using genetics. What did the MR analysis find?
It supported 454 potentially causal links between specific metabolites and diseases. Again, endocrinemetabolic and circulatory diseases were where most of these causal links were found.
454 causal links. Okay, based on this causality testing, which molecules emerged as the biggest definite risk factors and which looked genuinely protective?
On the risk side, a marker called XL-VLDL-PL% stood out. That's the percentage of phospholipids in very large VLDL particles. High levels of this were linked to a 5-fold increased risk of familial hypercholesterolemia.
5 times the risk. That's huge. A clear target.
Absolutely. And on the protective side, the most potent protective metabolite identified was SHDL-FC%, the percentage of free cholesterol in small HDL particles. Higher levels of this cut the risk for certain conditions by about 2/3.
Small HDL-free cholesterol. So protective HDL isn't just one thing. The type matters. That's really actionable information for drug development or lifestyle changes.
Exactly. And they took it one step further, connecting these metabolic findings back to the genes themselves.
That's so.
They found that for 402 of those potentially causal pairs, the metabolite and the disease actually shared underlying genetic determinants, a concept called colocalization.
Meaning the same gene variant influences both the metabolite level and the disease risk.
Precisely. They specifically highlighted SLDL-PL phospholipids in small LDL particles, which shared the most genetic signals with 9 different diseases. And they pinpointed a specific gene, PCSK9, and a particular variant, RS11591147.
PCSK9, the target of some major cholesterol drugs.
The very same. This variant seems to be the causal link connecting higher levels of SLDL-PL directly to multiple cardiovascular problems like familial hypercholesterolemia and coronary atherosclerosis. It perfectly validates why those PCSK9 inhibitor drugs work. They're hitting a genetically proven causal pathway involving these specific LDL particles.
Wow. This atlas really does cover the whole journey, doesn't it? From those 15-year early warnings in the blood to building predictive scores with machine learning and finally nailing down causal links right back to specific genes like PCSK9.
It's incredibly comprehensive.
It truly confirms that metabolic health isn't just some vague idea. It's dynamic, it's measurable, and it offers this highly predictive window into our future health trajectory.
And critically, the entire resource, the atlas itself, is open access online. Researchers everywhere can use it. Identifying these specific good guys, like SHDL FC%, and the bad guys, like XL VLDL PL%, gives us a concrete roadmap for developing much more targeted drugs and interventions.
Aiming to boost the protective factors and suppress the harmful ones.
That's the goal.
This whole deep dive really underscores that these metabolic signatures aren't just side effects of disease. They reflect deep, fundamental biological mechanisms we can actually identify and hopefully influence.
Absolutely.
So that leaves us with a really provocative thought for you, the listener, to consider. If this metabolic risk score, the MetRS, built from just the top 30 or so metabolites, is so powerful at predicting disease risk, sometimes even 10 years out, should regular monitoring of those key 30 markers become a standard part of our annual checkups, decades before symptoms might ever show up? Could this fundamentally redefine what we even mean by early diagnosis?
It's certainly something to think about. A potential future for preventative medicine.